in the description below. So I've got a lot of requests out there to talk about the new medication that's undergoing trials like remdesivir, and also vitamin supplements and, for instance, zinc, which we'll talk about shortly. To be able to understand how all of these things are affecting how the virus infects or replicates in the body, you're going to have some basic understanding of molecular biology. So bear with me over the next 10 to 15 minutes as we talk about the pathophysiology and the molecular biology of how coronavirus infect human cells. Now, in terms of nomenclature, remember that coronavirus is a family of viruses, and that family of viruses belong to a bigger group of viruses called nidoviruses. So you might see that term as well. I'm going to walk you through now exactly what happens with COVID-19 infection by the SARS-CoV-2 virus. We've got a cell. This side is the inside of the cell, and this is outside. And here we have the nucleus. And this is called the cytoplasm. The nucleus has a double-stranded set of DNA, which is deoxyribonucleic acid. And this holds all of the instructions for all of the proteins and structures and things in the cell. This is, in fact, the organism's genome. Now, what occurs next is something called transcription. And transcription is the copying of that DNA code into a single-stranded RNA code. And again, that's known as transcription. Now, that RNA molecule leaves the nucleus and goes into the cytoplasm, where it has a special modification that is made to it. And this special modification tells the cell that it's ready to be translated into a protein. And that is something called a 5' cap and a poly-A tail. In other words, a lot of A's at the end. Now, when that occurs, there's something called ribosomes. There's a large subunit ribosome and a small subunit ribosome. And that ribosome reads the code as it goes down from the 5' end to the 3' end. And as it reads the nucleotides 3 at a time, it comes up with a code that when those 3 letters are lined up, it means that there's a specific amino acid that has to be placed on there. And that uses tRNA, etc., etc. But the byproduct of that is you get a protein coming out of the ribosome. And of course, this protein is not made up of nucleotides. This protein is made up of amino acids. So this is a protein. And this is called translation. So that's the difference between transcription, which is going from a nucleotide to a nucleotide, and translation when you go from a nucleotide to amino acids to make a protein. And this is really what everything is made up of, is protein. Proteins have different structures. Proteins can be made into enzymes. Proteins can be made into all sorts of things in the cell. And that is what the cell does. And the codes for all of these proteins are included in the DNA. So this is really the central dogma of molecular biology. So now let's bring in the virin. I'm drawing lines here, but you should be aware that the outside of this cell is something called a lipid bilayer. And actually, the nucleus is a double layer of a bilayer, which we don't have to worry about. But if you were to look at this very carefully, there would be a hydrophilic layer on the bottom and the top, and in the middle, it would be lipophilic. So it's known as a lipid bilayer. Well, guess what? This virus also is made up of a lipid bilayer. And it has a hydrophilic side on the outside, and it has a lipophilic on the inside. So it looks very similar. This is the reason why you can destroy viruses with detergents. Detergents can break that up. This virus has proteins. Specifically, we'll talk about the coronavirus, since that's what we are concerned about. And there are proteins that are embedded in this lipid bilayer. And this protein that I'm drawing here right now is called the S protein, and it goes all the way through the lipid membrane. Now, there are other proteins in there. For instance, there are M proteins. There are also E proteins, and they all have their specific function of structure to keep this virus intact. And then what's inside is the genome. It's a tightly packed, very large RNA genome, and it's bound with N proteins. So there's N proteins all through here binding the RNA molecule. So you've got S proteins that help bind at the end here. They are going to dock with cells. You have an E protein, which is a membrane inside of the bilipid membrane layer. You have a M protein, which is a protein also in the membrane. You have N proteins, which help bind it. So all of these are proteins that have to be coded by something. Then, of course, you have the RNA. And that's key here, is understanding that coronavirus is an RNA virus. So in this situation, we have a receptor on the surface of the human cell here, which is known as the ACE2 receptor, or ACE2 protein. This S protein fits perfectly into that ACE2 and binds to it. And so when one of these comes and binds, what you get at that point looks something similar to this. And you can see here that all of the viral contents comes into the cytoplasm. Well, let's redraw this RNA molecule from the virus, and you'll start to see something very astounding. That's right. It's an RNA, and it has a 5' cap, and it has a poly A tail, just exactly as we have our messenger RNA ready for translation. Also notice that the cell incorporates the viral envelope as part of its outer membrane. And so as a result of that, these S proteins that were there initially now become part of the cellular membrane, marking it potentially for cellular destruction. Meanwhile, our ribosomes jump right on to this viral genome and start to march down the RNA. Now, this is what's known as a positive sense RNA. And what does that mean? It means that when the ribosomes come on to this RNA and they start to go from the 5' down to the 3' end, it's going to make a protein. And the protein that it makes is something called a RNA-dependent RNA polymerase. And so here it starts to make this protein. And there it is. And that's abbreviated RNA-dependent RNA polymerase, otherwise known as a replicase. And what this means is that this protein that is coded for by this mRNA will now take this positive stranded RNA, it's only one strand, and it will now march from the other end, because it reads from the 3' end, and it will start to march in this direction, going along the RNA, and make another RNA that is complementary to the previous one. Here, this will be the 5' end. This will be the 3' end. And of course, this is going to be complementary to the original, so this will be a negative stranded RNA. But guess what? This RDRP is then going to come onto this one and start over on this side, and it's going to read down this way. And the product of that is going to be the original viral genome. And so you can see here that this RNA replicase is going to be making more and more RNAs so that they can package these into new virons that this cell is going to be making so it can make more virus. And now the part that really blows me away, when this RNA-dependent RNA polymerase starts to go back on again, and it starts to do this, sometimes it doesn't reach right at the end. Sometimes it reaches from here and will only copy this part. And so you'll get a shorter RNA. Or it'll come from this side here and it'll get a shorter RNA. Well, when you get these shorter RNAs, these are known as subgenomic RNAs, or sgRNAs. And some of them are positive, and of course some of them are going to be negative because they're complementary. And this viral RNA-dependent RNA polymerase will just make more sgRNAs or negative sgRNAs. Well, guess what? These smaller ones also code for the smaller proteins on the virus. And so all of these RNAs can be seen and read by ribosomes that the host is donating to the cause. See, it hijacks your cell. And so these smaller proteins can be, for instance, M proteins, which the virus needs to be made for it. And it can also be E proteins. And so the virus comes into the cell, and the key here that you have to understand is it gives up its RNA into the cell, and it's ready-made for the ribosomes of the cell to make this protein right here called RNA-dependent RNA polymerase. Anything that ends in an ACE is an enzyme, and it tells you what it does. It's an RNA-dependent, which means it reads RNA, and it makes more RNA. That's what a polymerase is. It puts the little nucleotides together into a long chain with little beads. And that information helps tell the ribosomes, instead of making the proteins that your cell needs to make, it makes the protein that the virus wants to make, and it does it in this very clever way. So the question is, how can we interrupt this? Because what happens after this is some of these ribosomes are floating around in the cytoplasm, but some of these ribosomes are actually attached to a membrane which is contiguous with this outer membrane. And so they can insert all of those proteins just like they want, and then the reverse of what happened here at the beginning occurs, where these things bud off and little viruses get packaged and they go off. That's how one viron is able to go into one human being and millions of viruses come out. It's because it hijacks the molecular biology of the cells. And it happens to be that these ACE2 receptors are, generally speaking, in the lower portion of your respiratory tract. With all of these cellular outer membranes, with all of these foreign proteins, you know the immune system is going to start attacking these cells. And that's what happens with these lower respiratory cells. They become inflamed, they leak fluid, and that's what causes the pneumonia and the respiratory failure. So let's think about this. What would happen if we could somehow inhibit this nasty enzyme called RNA-dependent RNA polymerase? Well, as it turns out, there